All right, our next presenter is Amber Stilwell from the Regional Science Consortium, and she's going to be telling us about the research she's been doing with flow cytometry.
Hi, everyone. For those that don't know, I'm the laboratory manager at the Regional Science Consortium, and I had the pleasure of working with Mike and Rick for both my undergrad and my graduate work, so that was very, very fun. So today I'm going to talk to you guys about something that I've been working on all summer. It was also funded by Sea Grant, so thank you, Sea Grant, for funding this project. I use something called a flow cytometer to measure cell counts of cyanobacteria. So first I'm going to take you through what a flow cytometer is, because I'm pretty sure maybe no one in the audience knows what it is or is unfamiliar with the way it works. So this is the flow cytometer that I use. There are multiple different kinds, but this is the BD Accuray C6 Plus flow cytometer. And this is it as it sits on a desktop. And this is it on the inside. And I have zoomed in pictures of the inside later. So what the flow cytometer does is it uses red and blue lasers to collect a fluorescence signal from cells of algae. And this can be used for multiple different things. You can use flow cytometry to collect data on blood cells, red and white blood cells. And also I've read studies where they have collected samples and counted diatoms and all sorts of other things. So what I was particularly interested in was looking at the natural fluorophores that cyanobacteria give off as they go through these lasers. And those fluorophores include chlorophyll, which we've all heard of, but then two others, phycocyanin and phycoerythrin. So these cyanobacteria have these natural fluorophores, or a natural fluorescence. Phycocyanins are what are measured from our buoy. So like Dr. Diz showed the chlorophyll readings on the buoy. We also have a probe on the buoy that measures phycocyanin. So I also was able to measure phycocyanin in the cells using the flow cytometer. And this is all important because it goes through how I was able to actually obtain my data. So phycocyanins fluoresce at around 650 nanometers. So this list down here shows you all of the different light filters. And up here, there are 4 light filters on the inside of the flow cytometer. And those light filters essentially are picking up the fluorescent signals that are bouncing off of the cells as they go through the red and blue lasers. So to pick up phycocyanin, you have to use light filter FL4, and that is able to pick up wavelengths of 675 nanometers, plus or minus 12.5. And you can see FL4 here, and that is the filter, and then the fluorochrome that is actually inside the filter. And so we use the fluorochrome APC, and this will all be related later, I promise. It's a little to take in, or a lot to take in. The second important fluorophore is phycoerythrin. So they fluoresce at 488 nanometers, and so we had to use the FL2 light filter detector on the machine, and that is fluorochrome PE. So after reading a ton of studies and doing a ton of background research, I realized that the best way to look at microcystis as it passes through the lasers is by comparing the phycocyanin to the phycoerythrin, therefore giving me these axes of the light filters, PE versus APC. So these are a couple of the studies that I read showing the results that they found. So there are multiple variations of each fluorophore. And you can see up here all the different fluorophores that were analyzed in that one study. And then down here, this middle picture, you can see that the flow cytometer, the same model that we use, was actually used to collect data on different kinds of algae in the water, including diatoms and chlorophytes and cryophytes. So this piece of machinery can be used for many, many, many different things. It just depends on where those things fluoresce and if they have a natural fluorescence and if we're able to detect that. So down here is a study that I read that was able to look at Microcystis, Cyanotoxin, and Anabaena species all separately using the flow cytometer. And those axes that I mentioned before are here and here. So we have our phycoerythrin versus our phycocyanin. And you can see that all of these cyanobacteria are fluorescing in around the same area, but they all have varying mass sizes. So these are density graphs, and you'll see my particular density graphs later, but this is just showing you where that colony of, or where that species of cyanobacteria is fluorescing on your graph from your machine. So the machine is feeding all of that information to the computer model, and the computer model gives you this as it is being processed. So the density shows you the highest density of points of the cell. So that's where most of the cells are accumulating. But then you have these other cells around the outsides that may be dying off or may have a slightly different fluorescence than their the average cell. So this is a depiction, well, it's actually a screenshot of a standard of Microcystis that I ran through the machine. So after I figured out how to configure the axes, I was able to run a couple standards. Well, actually, I ran a lot of standards before deciding that this was the way to go. I also was able to get input from BD Biosciences, who created the machine, and they were able to tell me exactly how to gate around the colonies that I was looking for. And what that means is, so this here is everything. That is all of the data that was collected in my standard of Microcystis. And I had to adjust those axes to find what I was particularly looking for based on its fluorescence. So that's what this image is here. And you can see it's about, it's almost 100% Microcystis in my sample. And that makes sense because it's a standard. So from there, after you gate around this, you can actually label this and change the color so that it will show up in any of your other graphs. So this is a histogram showing one peak, and that one peak correlates to this, or corresponds to this coloring, this gating that I did around my standard of Microcystis. And like I said before, this is a density graph. These all three at the top are density graphs, and then these are population graphs. So they're just the number of events that occurred, and you can see the masses there in red and purple, and I'll get to what that red means later. So after I was able to figure out how to read Microcystis in the using the flow cytometer, and to figure out how many cells per milliliter of Microcystis were in each sample, that was my next step. So what I had to do was take all of my data, each sample I ran in duplicate, and they all went through the same process. You can adjust the settings here. You can change the size and the width of the needle that the cells are pulled up through the lasers by. You can also adjust the flow rate of the machine to change how fast it's pulling things through there. So after I figured all of that out, I ran every sample in duplicate. And after that, I would run a standard. And those standards, each one I would gate around, and then you can project that standard's location as it fluoresces onto all of your other samples and get percentages of the cell count, or of the cells that you're looking for. So from my original standard that I showed you guys, it was almost 100% Microcystis. I was able to run or to compare those cell counts or those gating areas to samples from, these are particularly from the Marina on Presque, and they're from the same sample, so Marina A and Marina B, so they're my duplicates. And we have about 7% Microcystis in the A sample and 6.9% Microcystis in the B sample. And I also wanted to show you guys both sets of samples just to show you how close together the machine is counting those cells. All of my percent CVs were very, very low, so I consider that very good. So the other part of this is knowing your debris. So environmental samples are dirty, and Barring a way to filter samples but keep my microcystis cells in them, I had to figure out a way to get the debris out of my samples before I can filter the samples. Because believe it or not, it's very difficult to filter microcystis out, or everything but microcystis out of a water sample, because microcystis is relatively large. So what you essentially have to do is look here in the very, very front corner of your sample. So this is my standard again, and this is that teeny tiny dot that's circled in red is debris. And I know that because when I can add these up together in the computer program, it tells me that this is, let's look exactly. I think it says 97.9% microcystis. So this would make up the rest of that chunk of the debris and what's not in my standard. So it could be dead cells, it could be different chemicals in the water that are being read by the machine, but we have to know what that debris is and where to look for it so that we can remove it from our analysis when we're looking at our dirty environmental samples. And yeah, I can also gate around that, and that I have in red. So if we look, this is my standard sample, and if you look at the histogram, that shows just the microcystis because the debris is actually so small, it's not showing up on our histogram. And for my Marina A sample, that is also projected onto here, and you can see all of those populations in red, all of that debris, and it does show up on our histogram here. But just a little bit of that purple there is our microcystis. But luckily, the machine allows us to batch analyze all of our samples so that we can actually only look at what we want to look at. So after I gate around that microcystis, this is the debris gating and this is the microcystis gating. I can choose to only analyze the microcystis graphs from all of my samples all at the same time. So if I select this graph and select all of my samples, then I get a long list of all of my statistics. But I can actually export all of that data to Excel or to PowerPoint, which is very, very convenient. And that was where I was able to take that data, pull it off the computer, and then do my post-processing to figure out how many cells per milliliter of microcystis that I have. So getting into my results, the whole goal of this is to try to figure out how cell counts of microcystis correlate to the microcystin toxin. Turns out that is extremely difficult, as you all probably know. So we analyzed the toxin by using the Abraxas ELISA kits, and we also did the quick lyse beforehand on all our samples. We did them in duplicate. And after the results were read by our microplate reader, we put them into the macro embedded data analysis worksheet, which is it's given to us by Abraxas themselves and helps us calculate the exact micrograms per liter of microcystins that we have in our samples. So then we can take those cell counts from the flow cytometer and compare them to the toxin concentrations that we got from the same sample on the same day. So these are our sampling locations. We actually had 24 sites around Presque Bay, along the Presque beaches, and on Lake Erie. This year we did something new. We actually extended our sampling locations all the way from Elk Creek towards the west, almost to the Ohio border, and all the way out to Freeport Beach, which is near the New York border. So all of these sites they enabled us to really broaden our database and see what's going on in more areas of Lake Erie, as well as all of our sites inside the bay and along Presque Isle beaches. So 6 sites along the beaches and then 12 sites in the bay. We also additionally processed water samples from Erie Waterworks and Northeast Drinking Water Authority. And Erie Waterworks brought us their drinking water samples, and Northeast Drinking Water Authority brought us their samples from Eaton Reservoir, as Dr. Campbell mentioned before, and also several other of their drinking water reservoirs, as well as their raw water intake from the lake. I should mention that we didn't actually do flow cytometry on the drinking water samples. We're hoping to have the funds to do that next year so that we can do cell counts versus the concentration of toxin. But for the most part, the samples from Erie Waterworks have very, very low concentrations of the cells and the toxins, so we didn't see the need to do that. I wish, looking at Dr. Campbell's data this morning, or this afternoon, I wish that we had done Eaton Reservoir because we would have had some really interesting cell counts to compare. So I said already that we sampled our locations, but we did it once a week, every Thursday, and we took the samples basically the same way that we take a bacteria sample. So you wade into the water waist deep, and then you stick your arms in elbow deep, unscrew your sterilized bottle, and then screw the cap back on and take it back on to the lab and make sure that it's chilled in the process. All of our samples were processed within 24 to 48 hours, depending on what staff we had and how much time we had at the end of the day. I mentioned that we did the quick lyse. We did each quick lyse sample in duplicate, and then we did our cell counts for cyanobacteria in duplicate as well. And we had 20 total sampling days. We actually did 21, but we only did 20 sampling days where we processed both toxin concentration and flow cytometry for the cells. And this is one of our interns, Julia Guerin, doing an ELISA test over the summer. She was learning, and she did a very good job. And this is one of our signs indicating that there's toxin levels above dog recreation threshold. So at that beach, or actually that's at Shades Beach in Northeast. So that day I took that picture. I was like, this is really good. I can use it for a presentation someday. So this was taken this summer. You can see it's very, very green. And I actually took this sample from the beach, believe it or not. Beach 6, we had a really, really bad bloom this year. And unfortunately, I don't have the data yet to compare. I have all the data, but I haven't sat down and compared the two from 2016 to 2017. But this was the first summer that we had a massive bloom on the beach side that was visible, lapping green waters up onto the lakeshore. So that day we had to go out and help the lifeguards determine whether or not they should pull people out of the water. Okay, so these are preliminary results, and the reason I say that is because a lot more work has yet to be done on this data set. So I'm just going to show you guys the flow cytometry results versus the microcystins results. So flow cytometry is indicated by the letters FC, and then MC indicates microcystins, the toxin concentration. So up here you can see the R data. So I did correlation charts for all of our samples, and I can answer questions about those afterwards. But these two in particular I wanted to show you. This was one of our highest correlations. So it's a pretty strong correlation. You can see the peaks moving up and down with each other at the marina on Presque Isle. But then at another location called Boaters Beach, you can see this peak here of the cells is completely opposite of the toxin concentration. So that's what's giving us our really low correlation. These are for the Presque Isle Bay samples. You can see all of our correlation values there. I was a little disappointed with some of these because places like Vista 3 always have really high toxin levels, but it didn't have as high of cell counts and not at the same time as indicated up here. And I think the reason for that may be because we actually collect that sample almost right in the morning, so at 7 a.m. or maybe 20 minutes after. And as Dr. Diz said earlier, they do have those gas vesicles where they can propel themselves up for the sunlight. So I'm thinking maybe that we have to change our methodology there so that we're getting sunlight exposure when we take that sample. And these are from the beach. We had some pretty strong correlations, especially at beach 11 was the only strong correlation. And then we have a very weak correlation at beach 2, which is interesting because that beach exceeded its values almost all summer for dogs. And then our East Erie samples, we had some high correlations at Freeport Freeport Beach. And at Mill Creek, we took this sample at the wastewater treatment plant location. However, we only have about 6 weeks of data for this, so it's a smaller data set. So I'd have to build that data set a little bit more next summer in order to consider this valid. And these are our West Erie samples, and as you can see, we did not have very strong correlations in West Erie, but at Elk Creek we did. We actually had an exceedance at Elk Creek one time for dogs, which that value is 0.2 micrograms per liter, by the way, so it's very, very low. That site was just very interesting because it's right where the water is very slow moving, and I think that it collects a lot of interesting things in it. So just to wrap up really quick, I know that I only have about a minute, but I think that one of the problems with our collection, maybe why we don't have such strong correlations as I expected, is that we only sampled once a week. So as you guys probably know, the cells can be there maybe on Wednesday, but then they're gone by Thursday when we take the sample. So we're scooping that water, we're taking the sample and analyzing it, and we're getting those high toxin concentrations, but not necessarily high cell counts. And then I also need to look and see at, the comparison between the other toxins that we analyze, which are anatoxin, saxitoxin, and cylindrospermopsin, because microcystis can release multiple different toxins, but my other issue is that the flow cytometer right now, as I'm running it, cannot differentiate between cyanobacteria. So I did a couple trial runs before the summer started. I combined Anabaena species with microcystis, and it did not look any different. I was not able to find a density that was significantly different than the other. So that's something that I'm going to work on this year, and trying to run different types of cyanobacteria through there and see if I can get a better handle on splitting those genera apart, and hopefully that will enable us to have a better ability times with our cell counts if I can get us there. So again, thank you to Pennsylvania Sea Grant, Great Lakes Restoration Initiative, and the PADEP, specifically the Coastal Resource Management Program, for funding this research, and also to BD Biosciences for helping me analyze some of my data, and to Jeanette Schnars for supporting this project, and also to all of our interns who worked really, really hard this summer to collect all the samples. And any questions? I know.
So one of the, it's great, you saw some really good correlations. Do you think that the fact that you're lysine cells has any effect on the correlations that you're seeing? So if you just looked at extracellular toxin, do you think that some of those that you didn't see correlations that might tighten up?
Yeah, that's a really good point. We should definitely look at doing that for next year, because yeah, you're right. If we are lysing all the cells, we're getting our maximum toxin concentration. So yeah, that might not necessarily be, yeah, thank you.
I can't remember, was there a pattern, was your cell count always lower than your toxins when you had those, when you didn't have correlation?
Mm-hmm.
Was there one always higher than the other, or was it a mix?
It was kind of a mix, yeah. Some of the correlation graphs show really, the patterns are moving together. Generally, I think the cell count was higher than the toxin concentration, just by looking at those, because obviously they're on different graphs. Yeah. But at some points, we did see the toxin concentration exceeding the cell count. But then other times when, we had zero correlation, like even a negative correlation, it was, high cell counts 1 week, and then at the end of the summer, high toxin concentration, and just, yeah. So I need to look more at the general area. I want to look at the general characteristics of those areas to see maybe if they're shallower or if they have warmer temperatures generally, because we collected a ton of other data that I just have yet to dig into.
I was just wondering too, if it's sort of like almost kind of opposite, like if it's a legacy, where the cells have moved through, but the chemistry is remaining or something.
Right, yeah. And we do have some nutrient data that we collected from the stream locations that we process. So hopefully we can get that analyzed, and I want to make that comparison as well and look at that to see. Did you?
Well, tagging on to those ideas, if you're on the leading edge of a bloom, then perhaps, total speculation, you've got a lot of young cells, relatively speaking, that have not lysed or exported toxins to the water. If you're on the trailing edge of a bloom, you might have a lot of old cells that are coming apart and releasing the toxin to the water. So you could have the same number of cells on the leading edge versus the trailing edge and have a very different physiological state for those cells. Does that make sense?
Yeah, it does. Yeah, and I want to, if we can, someday sample more than 1 day a week so that I can try to compare, you know, if maybe on Wednesday, you know.
You're already.
I know, and I may be even able to look at last week compared to this
Did you
week.
study with Dr. Campbell?
Yes, I did.
I can tell.
Okay, thank you.